All right, so I have another cool science news story for you here. Posted in Real Clear Science. This animal has near perfect regeneration, and it's not a salamander. When it comes to regeneration, salamanders, and particularly axolotls, garner lots of attention. The rather adorable critters can completely regrow lost appendages dozens of times, and to a lesser extent can even restore their eyes, heart, and certain parts of their brain. But when scientists have taken a closer look, they found imperfections in the regenerated body parts. Axolotls are good, but they're not that good. Now, this reminds me of a study I covered, I think, last year, might have been 2 years ago. But basically, there was a drug cocktail and a new like treatment device that researchers were testing to regrow limbs, and they were testing it on frogs. They would amputate a frog's limb, attach this device to the stump. The device would inject or expose the stump to this cocktail of drugs and medicines. And the whole idea was they were trying to recreate like the conditions in the womb so that when the limb regrew, it would follow more natural, like embryological developmental pathways. And they were able to, to a degree, successfully recreate that. And the regenerated limb had a lot of details that they didn't expect. Like they didn't expect to get a complete full recreation of the entire limb with like muscles and bones and nerves and the whole 9 yards, but they got more than they thought. If I recall correctly, there was nerves and bones, just not in the toes, and some of the toes didn't form completely. I think I'm remembering that correctly. I could be wrong about some of those details, but in any case, a big concern was the completeness, the perfection of the regeneration. You're not going to get a perfect regeneration if a limb is completely severed. And so here in this article, they just mentioned that about axolotls and salamanders, but, and this is the topic of today's science news here, of this important discovery, Parhial hawaiiensis, on the other hand, does seem to be that good. A team of French and German scientists recently severed legs from a few of the shrimp-like crustaceans and watched over weeks and months as they regrew with near-perfect fidelity. The team's findings were recently published in the journal Science Advances. Parhiale legs are quite complex structures, featuring numerous types of sensory organs called setae, which the organisms use to feel their surroundings. So that's these things you can see in the image here. They're like stiff hairs. Setae are composed of a few cells and take different shapes depending on their type and function. Their complex and stereotypic external morphology enables easy detection of even minor defects, the researchers wrote. So one of the details they were comparing here for the completeness of the regrowth of the limb is the presence, shape, orientation, positioning, etc. function of these setae. The scientists found that after 3 months, regrown legs were identical in size and shape, with indistinguishable setae in the exact same locations, and each had the same sensitivity as before. They also probed cellular composition in the legs and profiled cells at the molecular level, finding no differences whatsoever between the animal's original and regenerated legs. While we cannot exclude the existence of subtle patterning or functional defects in regenerated Parhiale? I don't know how you pronounce that genera name. Parhiale legs, at the current level of morphological and molecular resolution, these legs appear to be perfect replicas of the original legs before amputation, the scientists concluded. Well, that is just super cool stuff. The implication here is that if some enterprising scientist could find out, a genetic pathway or a drug or something that could that's responsible for this, for the super high fidelity of this regeneration, you could theoretically translate that into human medicine and theoretically cure amputees. And that's like one of the many holy grails of medical science, is if you can develop a technique to perfectly replace like someone's severed leg or their severed hand or something. Say they suffered a trauma in a car accident or an industrial accident, or a violent injury from self-defense or something. Being maimed like that has a huge consequence on their quality of life. But if the limb could be, cleanly amputated, cleaned up, and then regrown to perfect fidelity, I mean, that would be revolutionary. That would be an amazing game changer. So let's check out the article here, the scientific paper in Science Advances. Crustacean leg regeneration restores complex microanatomy and cell diversity. Abstract. Animals can regenerate complex organs, yet this process frequently results in imprecise replicas of the original structure. In the crustacean Parchiale, embryonic and regenerating legs differ in gene expression dynamics, but produce apparently similar mature structures. We examined the fidelity of Parchiale leg regeneration using complementary approaches to investigate microanatomy, sensory function, cellular composition, and cell molecular profiles. We find that regeneration precisely replicates the complex microanatomy and spatial distribution of external sensory organs and restores their sensory function. Single nuclei sequencing shows that regenerated and uninjured legs are indistinguishable in terms of cell type composition and transcriptional profiles. This remarkable fidelity highlights the ability of organisms to achieve identical outcomes via distinct processes. Okay, so just to kind of go through this again, the complex microanatomy and spatial distribution of external sensory organs and restores their sensory function. So not only do you have the physiological mapping, like where does this stuff go, where does it grow out from, but you also have innervation of these organs and structures and tissues. The sensory organs, they feel, they report data back to the brain, back to the central nervous system, and you see that with the restored sensory function. The animals would then, respond to sensory inputs using those nerves. And then single nuclei sequencing shows that in terms of cellular composition, so what types of cells are in the regenerated limb, and transcriptional profiles, so this is like what kind of genes are being expressed, what kind of proteins are being produced, that kind of thing. It's pretty much indistinguishable from a normal, healthy leg that was never regrown. This remarkable fidelity, yeah, that's an understatement, this is amazing, highlights the ability of organisms to achieve identical outcomes via distinct processes. So what this means, identical outcomes, they're producing the exact same limb, but distinct processes. Well, originally the limb was created through embryological developmental processes, molting and metamorphosis and things like that are necessary components of the animal's life cycle. But the regeneration, that's its own distinct process, separate from just the natural growth cycle. This regeneration process is different, but it produces the same organ, the same identical outcome. And that really is amazing. Okay, so this is actually a pretty brief paper, it looks like. Introduction, results, talk about the results a fair bit. Just looking at some of these, the graphics that they have. To assess the robustness of our analysis, we tested different methods of data integration and different read mapping strategies. Okay, so you like to see stuff like that, where they're like double, triple, quadruple checking their stuff, like coming at it from different angles and testing their data from different perspectives. That's good to see. Nice. So yeah, they organized their data in different ways, trying to see if they could parse out any patterns or any differences, and they didn't find any. Okay, so yeah, let's wrap up with the discussion here. Overall, our data reveal that the regenerated legs of Parchiale are indistinguishable from uninjured legs. This conclusion is based on complementary approaches interrogating the microanatomy, sensory function, cellular composition, and molecular profiles of cells. While we cannot exclude the existence of subtle patterning or functional defects in regenerated Parchiale legs, excuse me, at the current level of morphological and molecular resolution, these legs appear to be perfect replicas of the original legs before amputation. Our approaches for probing morphology, sensory function, cell diversity, and molecular profiles have some inherent limitations, which are important to acknowledge. Okay, so we can go through this. This is good to know. This is just the limitations of their study techniques, the limitations of the methods they used, and it puts some constraints on how far they can interpret this data. And this is a really important part of science, so I should mention this. They say, on the microanatomical level, although we conclude that leg regeneration in Parhiale restores the full pattern and diversity of external sensory organs at the tips of T4 and T5 legs, we have not probed other aspects of morphology and microanatomy. For example, patterns of internal proprioceptive neurons, so neurons that tell where the limb is in space, like, oh, my leg is raised, my leg is down, legs crunched up, legs extended, that kind of a thing. As well as muscles, tendons, or muscle innervation. Using a behavioral assay, we show that regenerated mechanosensory organs are sensitive to touch, but we are not currently able to probe how sensory signals from these organs become integrated in more complex behaviors. On the cellular level, we are confident that all the identified cell types are recovered during regeneration. If any major cell type was missing, such as the missing bone cells and regenerated lizard tails, then our approach would have uncovered that difference. We observe no significant differences in the relative proportions of different cell types between control and regenerated legs, but we acknowledge that with the observed levels of variation between samples, the sensitivity of our approach is limited to detecting greater than twofold differences in the abundance of any given cell type. Similarly, although we do not observe significant differences in the molecular profiles of cells in control versus regenerated legs, we recognize that probing differential expression in single nucleus studies has inherent challenges. For instance, a relatively low sensitivity. Okay, so I want to just go over some of this stuff because it's important. One thing they say is that they did probe mechanosensory organs that are sensitive to touch, so they would have touched the limb, a mechanical sensory input, and seen how they respond. But what they're saying is that that's about it. And in terms of the neurological response and the neurological function of this regenerated limb, they don't have too much more knowledge. They don't know if it's able to engage in more complex behaviors, like if it allows them to walk in a coordinated fashion, or if, for example, they mentioned that they didn't probe those proprioceptive neurons. So it's possible that this regenerated limb might have deficits in its, the sensory feedback telling the organism where the limb is in space, or perhaps there's some aspect with like pain or temperature sensing or something like that that has suffered some sensory feedback disruption. They're saying here that their analysis wasn't that detailed, that in depth to pick up those kinds of details. Also, another big thing here is the sensitivity of our approach is limited to detecting twofold differences in the abundance of any given cell type. So what this is saying is like say you have cell type A and cell type B, and both of those cell types are present in the original and regenerated limb types, but let's say cell type A in the original was at 2/3 the frequency that you see those cells in the regenerated leg. So say it gets overexpressed a little bit more in this regenerated leg. They wouldn't have been able to pick up a difference like that unless that overexpression was at least twice what it was in the original leg. And so that's kind of a big difference. And it's a bit of a bummer because it'd be interesting to know if there was smaller differences between the leg. Like say the regenerated leg overexpresses by 15% this cell type, or the regenerated leg underexpresses some kind of connective tissue producing cell type by 20% or something like that. Those would be really interesting little details, little variations between the original and the regenerated limb, but they're saying here that their analysis wasn't fine enough to detect a difference below 2 times the ratio. Well, anyway, the remarkable fidelity that we have observed in Parhiale raises interesting questions about the mechanisms underpinning faithful regeneration of complex organs. Previous studies have suggested that the same regulatory networks are deployed to build an organ in embryos and in regenerating adults. Recent work in Parhiale in the sea anemone Nematostella brachiale, and in the sea anemone Nematostella vectensis, however, shows that development and regeneration have different temporal profiles of gene expression. We suggest that development and regeneration could be driven by distinct global regulatory mechanisms, yet incorporate some shared genetic modules for patterning and cell differentiation that lead to a conserved outcome. Well, very complex stuff, but very, very fascinating. Now, I would be really interested to see if they could, build on this, dig a little deeper into this, find out what exactly is going on, how they do this, and then see if you can apply that to vertebrates or mammals or humans. Because, yeah, like I said, that would be a game changer.